Okay, so today I'm going to talk about the liver metabolism for our last colloquium. I just want to say that I'm a student and I'm doing this as I learn, so if I say anything wrong, just tell me and I'll try to correct it either in the comments or in the description below the video. So first and foremost, the main functions of the liver, first of all, is metabolism. Metabolism of the carbohydrates, proteins, and lipids. The second thing is that the liver produces plasma proteins, and we will talk a lot about plasma proteins in this lecture. The third thing is endocrine function of the liver, in which it will release hormones or hepatokines. FGF21 is such a hormone that will increase insulin sensitivity of the cells around it, or like in the human body, and also it will increase glucose uptake. The liver also has exocrine function, in which it will produce bile and then secrete it into the gallbladder. It also has storage function, in which it will store proteins. It will store, it will store fatty acids and also glucose into glycogen form. It will also store vitamins A, D, E, and K, and vitamin B12, and hormones as well. Another function is detoxification of alcohol and drugs, but also detoxification of ammonia and converting it into urea, because urea cycle only happens in the liver. Another function would be I don't think we have to know this exactly, but how it works, but the liver has Kupffer cells that will get rid of the erythrocytes. So right now in this lecture, there's a lot of repetition from our last colloquium, so I will just go through that very quickly. So how does the liver control glucose homeostasis? And in one word, it will be glycogen. So what I mean by that is the liver can control glucose homeostasis by two hormones. It's insulin and glucagon, and these two hormones will regulate glycogen. So if we have insulin, that means that we have just eaten and insulin will be produced. Therefore, the liver, what it will do is actually store more glucose, because we have glucose now in excess, and so insulin on the liver, it will act on the GLUT2 receptor, and it will activate glycogenesis. So not GLUT4 receptors, they don't exist in the liver, and the liver is insulin independent. So that's important to remember. Insulin will decrease gluconeogenesis, and it will decrease glycogenolysis. So if you have to remember one thing, is that insulin does uptake and storage, mainly storage of glucose in the liver. And the glucagon will do the opposite of what insulin does. Another thing that we have to know is glucose 6-phosphate's importance, and glucose 6-phosphatase, what they are and what they do. So in this schematic picture, what we have to remember is there will be glucose in the blood, and it will be uptaken by the liver, and then it will be converted to glucose 6-phosphate, which is this molecule. And in order to do that, you would have to have the enzyme hexokinase. Yes, but if you want to get glucose out to the blood, then you would need glucose 6-phosphatase that will break down the glucose. It will take the phosphate away from the glucose, and in that way the glucose can go out to the blood. Another thing that's important, so glucose 6-phosphate is important because it's what starts multiple different pathways. Well, it is needed so that many different pathways can start. So in order to do glycogenesis, you would also need glucose 6-phosphate. So that's when you go to here, or when you start a pentose phosphate pathway, you would also need glucose 6-phosphate. Yes, but that is so we will do glycogenesis in case we have insulin. But if we have glucagon, then we will do glycolysis, and then we will get pyruvate, and pyruvate will then be converted to acetyl coenzyme A, and then we will go through the citric acid cycle, and then we will get energy. That is in the case of glucagon. But if we have insulin, then what we need to do is we need to store our acetyl coenzymes A, because we have already a lot of ATP, and this will inhibit the enzymes in the citric acid cycle. Therefore, we will get a lot of or we will accumulate a lot of citrate, and we want to store the energy instead. So when we have a lot of citrate, this will allow for citrate transporter, and we will get citrate outside, and then citrate in the cytosol. So this is the cytosol. In the cytosol, the citrate will be converted into acetyl coenzyme A, and then we can make cholesterol synthesis, fatty acids, and tags and phospholipids. And so how does the liver regulate lipid metabolism? Kind of in a similar manner. We have 3 important hormones. So here we have the 3 important hormones in this case are insulin, glucagon, and thyroid hormones. So in the case of insulin, we will make liver lipids. So as you can see here in step number 1, we will make liver lipids. That's if we have glucagon, or if we have the thyroid hormones, then we will do beta oxidation. We will get acetyl coenzyme A. We will make cholesterol and bile salts if we have insulin present. Yes, but in case if we have glucagon present and the citric acid cycle is not working because not enough intermediates, because we don't, for example, a low carbohydrate diet, then we will make ketone bodies. Yes. Also here, the plasma lipoproteins will be produced in case you have insulin, and the free fatty acids in the blood, that will happen in case of glucagon, or in case of T3, T4 thyroid hormones. So how does the liver regulate amino acid metabolism? Also, the important hormones here would be insulin, glucagon, cortisol, and epinephrine. Insulin, in case of insulin again, we will make liver proteins, we will make hormones, porphyrins, nucleotides, and in case of glucagon, cortisol, and epinephrine, we will try to make energy. Therefore, for example, alanine that will be broken down from the muscle will be through transamination make pyruvate, and then pyruvate will be glucose, and glucose can go out, and we can do glycolysis in the muscle. Well, pyruvate to glucose is gluconeogenesis. Another important thing now when we convert alanine to pyruvate is we can't forget about the urea cycle because of the NH3 that will be produced. Yeah, that's all I had. Oh yeah, and this is also a reminder that in transamination reactions, you will go from, like, for example, alanine to pyruvate, you will produce ammonia, or from glutamate to alpha ketoglutarate, you will also produce ammonia. How does the liver act in a well-fed state? And that means that we are in a lipogenic state, meaning that we have insulin present. If we have just eaten, so if we take a look here at the intestine and we have just eaten a meal, then in that meal we have glucose, amino acids, and fats. And how does the liver act in this state where there is insulin? So insulin will tell the liver to do glycogenesis, because insulin will act on the GLUT2 receptors, and the GLUT2 receptors will signal to do glycogenesis. Therefore, glucose will be uptaken through the blood vessel and then to the liver, and then glycogenesis will happen. What will happen then to the amino acids? Amino acids will also be uptaken by the liver, and they can either become protein, so protein synthesis will happen, or we will make alpha keto acids, and these alpha keto acids will be converted to acetyl coenzyme A that later then is stored into the form of tags. Because alpha keto acids, you could make energy, but in this case, because this insulin is present, we will make tags. So that's that is highlighted by number 5. Number 6 is these tags will then be together with apolipoproteins. We will form lipoprotein VLDL that is then transported to adipose tissue, and we will store the tags here. They will also be stored in muscle, but mostly in the adipose tissue. And yeah, that's that about that. How does the liver act in fasting state? And in that case, glucogenic state. So in that case, glucagon is present. And if glucagon is present, then it will signal to the liver to do glycogen breakdown. Because when doing glycogen breakdown, we will get glucose 6-phosphate, which once again was that important enzyme. And glucose 6-phosphate is then converted to glucose by glucose 6-phosphatase. And then it can go out, and then it can go to the brain. Also, when glucagon is present, proteins can be broken down. Amino acids go to the liver. So protein both in the muscle and protein in the liver can be broken down. And amino acids can then be formed into pyruvate. So for example, alanine will be formed into pyruvate, and then gluconeogenesis would happen, and glucose can go to the tissues that need it. Amino acids can also be formed into ketone bodies, and in that case, ketone bodies can go to the brain, for example. Acetyl coenzyme A, I'm getting ahead of myself. So that will happen. And now in number 4, adipose tissue, when glucagon is present, will release, will activate lipolysis. Glucagon will activate lipolysis, hydrolyzing the tags and forming glycerol and fatty acids. And in that case, the fatty acids will undergo beta oxidation, and they will produce acetyl coenzyme A. Acetyl coenzyme A can be formed into ketone bodies or go through the citric acid cycle to produce ATP. Also, this is here. I think I just copy pasted what was written on the slides, but or maybe I didn't. I don't remember. But quickly, this is a repetition from the 3rd colloquium. Inborn metabolic diseases, fructose and galactose, and glycogen storage diseases. So the 2 fructose diseases that we have is when we have a deficiency in fructokinase, which is a disease that really is benign. It doesn't do anything. The person will just pee the fructose out. The second disease is a deficiency in aldolase B, which is also called hereditary hereditary fructose intolerance. And in that case, the phosphate right here will not be released. And this is really bad because in electron transport chain, we need a lot of inorganic phosphates to add to the ADPs so that we can get ATP. The problem though is when a person has this disease and like aldolase B is not present, then the phosphate will not be released and therefore there will be no inorganic phosphate to make ATP, and this is not good. And glycogen storage diseases is basically when there are many different types, but an example would be the von Gurk disease, in which a person lacks the ability to get the glucose out of the cells. So for example here, if a person has von Gurk disease, the glucose molecule right here can never exit because the person lacks glucose 6-phosphatase enzyme, and therefore glucose 6-phosphate is never converted to glucose, and therefore the glucose can never go out into the blood. So that would be an example of a glycogen storage disease. And then we have galactose disease, which could be, for example, galactokinase deficiency, in which galactose is not metabolized. Therefore, we will have another enzyme, aldolase reductase, that will form galactitol, which is harmful. Yes, okay. Now we are done with the older colloquiums. What is the main function of plasma protein and the main types, and where are they produced? So plasma proteins, as I said in the beginning, they are produced by the liver. And there are different types of plasma proteins, and they have different functions. So the most common one is albumin, and albumin does two things. It transports fatty acids and also nutrients, and it maintains the osmotic pressure in the blood vessels. Then we have alpha globulin, we have beta globulin, and we have gamma globulin. And they also have different functions. So alpha globulin, it transports vitamins, and alpha-2 transports hemoglobin. Then beta globulin, there are three types of beta globulin. So transferrin, hemopexin, and fibrinogen. And I think we have to know this one. Oh, we have to know this one for physiology. And I think we have to know all of them, actually. And then gamma globulin is antibodies. And my friend, I always used to mix up antibodies and antigens, but my friend told me that antibodies can be shortened to ABs, and you have ABs to protect yourself. So antibodies will be these proteins in the body that will help you against the antigens. And antigens are, you could think of them like as spikes on a dinosaur, and your antibodies will attack these antigens, these spikes on the dinosaur, and it will protect you. Okay, and then we have C-reactive proteins. C-reactive proteins will increase during inflammation, and especially in bacterial infections, they will increase by 2,000 times. So really, they are a really good marker, and this is very important to remember. They are a really good marker for bacterial infections, and this is something we have to know. And then we have complement factors. So when an antibody binds to an antigen, a complement factor will see this, and then it will go and attach itself to that complex, so that it will flag to the macrophages that to come and eat this antigen. And then we have coagulation factors, which are also plasma proteins, so factor 1, factor 2, etc. Yeah. Now we're going to talk more about alcohol. But first and foremost, what two pathways are anaerobic carbohydrate pathways? We have the ethanol pathway and the lactate pathway. Ethanol pathway is when, and it can only happen in yeast, and that is when glucose is converted to ethanol. But that does not really happen in, and this is like minus oxygen. There's no oxygen, that's why ethanol forms. And the same thing happens in humans. Well, not the same thing, but what can happen is glucose is always converts to lactate in case of anaerobic conditions. So why does that happen? And this is something that we really have to know for our colloquium. Why does glucose get converted into lactate? Lactate really isn't the main goal. The molecule itself is not really used, but what we're interested in is the 2-NAD+ that is formed when forming lactate, because when we go from glucose to lactate, we will form these 2-NAD+. These NAD+ will then be used in the 6th step of glycolysis. In this step, we need NAD+, that will then be converted to NADH. And the NADH we need, with the NADH we won't be able to use in anaerobic glycolysis, but in order for the 6th step in glycolysis to happen, we need to have NAD+, because when we have the NAD+, we can then do the rest of glycolysis, and we can get pyruvate, but the main goal is actually the 2 ATPs. So yeah. Now to oxidative alcohol metabolism. How does the body get rid of or neutralize alcohol? And where does that happen? So that happens in the liver. So ethanol enters the liver. Ethanol enters the liver, and by alcohol dehydrogenase, we will form acetaldehyde that will then diffuse. Acetaldehyde gets formed, and then it diffuses into the mitochondria. But as we form acetaldehyde, we will actually form NADH, which is an important thing to remember. Then in the mitochondria, acetaldehyde is actually very toxic, and it needs to be neutralized. We don't want it to go out into the bloodstream. Therefore, we will convert acetaldehyde to acetate. And as we do that, we will produce NADH. Now, the acetate that we have formed can diffuse out and go to the muscles, for example. And then from acetate, we can form acetyl coenzyme A, and that can go through the citric acid cycle, and we can get energy. Yes. Now, the conversion of ethanol to acetaldehyde can happen in 3 different places. It can happen in the cytosol, which is the most common place for it to happen. Like, I think 80%, 90% of alcohol metabolism or conversion of ethanol to acetaldehyde happens in the cytosol. And it happens by this enzyme, alcohol dehydrogenase. Okay, and then it goes to the mitochondria and it forms acetate. The other place is peroxisomes, and in this case they won't use alcohol dehydrogenase, but they will use catalase by using hydrogen peroxide with the ethanol, and it will form acetaldehyde. and water. And then the acetaldehyde will diffuse into the mitochondria. The third way is in the microsomes, so an organelle in the liver, in which it will use NADPH and the ethanol. So the enzyme right here will use NADPH and ethanol to convert it to acetaldehyde. And this happens, so 10 to 20% of alcohol metabolism will be used in case of chronic alcohol abuse. Oh no, in 10 to 20% will be in normal case, but in case of chronic alcohol abuse, then that will increase by 4 to 10 times. Now, this is really not good because we will deplete our NADPH stores if we deplete NADPH. This is not good because then we cannot neutralize reactive oxygen species that really, like, NADPH is used to neutralize this, but also in order for fatty acid synthesis, we also use NADPH. So both of these things will not happen, and this is not very good. Yes. So why is alcohol toxic? There are two reasons. There is NADH accumulation, but also acetaldehyde accumulation, which I will talk about in the next slide, which has to do with this one and the microsome alcohol oxidation. So, but not right now. I'm just going to talk about this in this slide. So, as we convert ethanol to acetaldehyde, we will produce a lot of NADH. And then it will go to the liver, where it will go to the mitochondria, where it will be converted into acetate. And now that there is a lot of NADH, when there is a lot of NADH in, let's say, for example, an alcoholic, this will disrupt the balance between NAD plus and NADH. If we have an excess, we will get an excess of NADH. This will inhibit the citric acid cycle. If we inhibit the citric acid cycle, the acetyl coenzymes A will accumulate and they will form ketone bodies, and we will get ketoacidosis. So that's one thing that will happen because of the NADH imbalance, NAD plus NADH imbalance. Another thing that will happen is that the body now wants a balance between these two, right? So glycolysis will happen at a much higher rate to form pyruvate and then later form lactate. Why? Because when we form lactate, we will form NAD plus. And in that case, that's an attempt for the body to try to neutralize the NADH, NAD plus balance. Also, so this will lead to lactate acidemia, but it will also lead to hypoglycemia in the body because alanine and other gluconeogenic precursors will be converted to pyruvate, and then once again, in order to form NAD plus and neutralize all of this. Now, this is really not good to form a lot of lactate because in that case, lactate will compete with the uric acid to be excreted with the urine. And in this case, uric acid will actually accumulate a lot and form gout. So, and purine metabolism will just happen naturally, and because lactate will be at a much higher concentration than uric acid, then that will be excreted in the urine and not and not uric acid. The other thing that will happen is hyperlipidemia, and this will happen because once again, to try to neutralize the NADH and NAD plus balance, we have a lot of NADH and we want to get NAD plus. Therefore, glucose glycolysis will happen. We will get dihydroxyacetone phosphate. We will get glycerol. Once we have glycerol 3-phosphate and we have fatty acyl coenzymes A, because we have a lot of NADH, we don't need to do beta oxidation. So beta oxidation will not happen. Instead, we will store fatty acids. And so now that we have fatty acyl coenzyme A and we have glycerol, we will make a lot of tags. They will be stored into VLDLs, and therefore we will get hyperlipidemia. And then why is alcohol toxic? So the acetaldehyde accumulation. Acetaldehyde is highly toxic, and it will form Schiff-base adducts. And these Schiff-base adducts will want to steal electrons from proteins. And by doing that, some certain proteins will not be able to be functional, or they will not be able to be excreted out. So that's one thing that acetaldehyde can do. Also, if somebody is, for example, an alcoholic, the microsomal alcohol oxidation will happen at a much higher rate, meaning that the NADPH stores will be depleted. And that means that the reactive oxygen species will not be able to be neutralized. And in that case, we will have another antioxidant that will be depleted, which is glutathione. Because glutathione will also donate its electrons to the reactive oxygen species. But if both NADPH and glutathione are depleted, then really there will be also a lot of reactive oxygen species that are just running around in the liver, and that's not good, as it will steal electrons from the phospholipids and from the proteins and DNA. Another problem with this acetaldehyde is that it will actually bind to these proteins. For example, apolipoproteins are needed in order to make these VLDLs so that the triacylglycerols can leave the hepatocyte. But if the acetaldehyde is present and in high amounts, then it will bind to these proteins, and the apolipoproteins and the lipoproteins will not be able to be formed, and VLDL will never leave the liver. So it will never leave the liver, and this will lead to fatty liver disease. Also, so a biomarker of this is we can actually see that ALT and AST will increase in the cell, will increase in the blood, because a lot of hepatocytes will rupture and die because of this, because of the toxicity. Then we have Xenobiotica metabolism. So there are two phases. We have phase 1 and phase 2. In phase 1, when we have a xenobiotic, we have some sort of R-chain, doesn't mind how it looks like, but we have a hydrogen. The first step would be to make this more water soluble, and that will be the phase 1 reactions. And in the phase 1 reactions, we will use CYP450 enzyme. And this enzyme has two components. The first component would be cytochrome P450 reductase, which is this one, which is when FAD gives it receives electrons, and then it gives them to FMN, and then FMN gives them to cytochrome P450, and then this cytochrome P450 can donate electrons to the Xenobiotica, or to any drug or anything like that. Then phase 2 is the actual detoxification phase, and it is when we conjugate the more water-soluble form of the drug, We can do this by these 4 different things. So glucuronidation by this enzyme, and sulfation, glutathione conjugation also, and acetylation. Another important thing that I wanted to say about the P450 is actually that their activity will increase by having more substrates. So if there is more drugs or xenobiotics that need to be metabolized, then P450 transcription will increase and P450 will increase, and therefore we can metabolize the drugs. And now toxicity of drugs, and it's kind of very similar to toxicity of alcohol. What you just need to know is, for example, if somebody overdoses on paracetamol, this can be deadly. Why? Because paracetamol will enter the liver and then it will be metabolized by this enzyme. But what will be formed is this one. This is really important, NAPQI, which is highly toxic and it can lead to cell death. But it is neutralized by glutathione. The problem is, if somebody has overdosed, this means that all of the glutathione will be used. And in that case, there is no more glutathione and no more antioxidant that will be able to neutralize the amount of NAPQI, and this will lead to cell death of the liver, of hepatocytes. Now, what causes jaundice? You have to just know and remember one thing, that bilirubin, so it is a high amount of bilirubin in the blood, which is a yellow pigment. That's why jaundice is when somebody is kind of very yellow. But let's talk about that. So in a normal case, in normal cases, let's say this blood cell dies. And as it dies, it will release heme, right? And heme will then be converted to biliverdin, and then it will be converted to bilirubin. Bilirubin will then be uptaken by the liver, and then from the liver it will go to the gallbladder, and then from the gallbladder it will go to the guts. And then in the gut, in the intestines, it will be converted from conjugated bilirubin to urobilinogen, in which it will be excreted with the urine or through the feces. That is the normal case. Now, there are 3 ways in which this will be obstructed and cause jaundice. So the first kind of jaundice would be prehepatic jaundice, in which there is nothing wrong with the liver or with the guts, or there's nothing wrong there. The problem is that there is just a lot of hemolysis happening at one time, a lot. Therefore, there will be a lot of unconjugated bilirubin in the blood. The problem is that the liver will be overwhelmed by how much bilirubin it needs to conjugate. It will conjugate a lot of it, but not everything. So the person will still look yellow. But the conjugated bilirubin will then go to the gallbladder and then to the intestines, and we will get a lot of urobilinogen, and that will be excreted through the kidneys. That's also one thing. Remember that bilirubin is not water soluble, so it will never be able to travel all the way to the kidneys and be excreted with urine. It cannot do that. It needs to be conjugated. Okay. Intrahepatic jaundice can be So as you can see here, it's not because of excessive hemolysis. Yes, it is hemolysis, but the problem here lies within is because of the liver. Oh my gosh. Like, I don't know what's happening. It's because of the liver. So the problem here is that there will be some bilirubin uptake, and the liver will conjugate some bilirubin, but not everything. It cannot take up everything. The problem here with the intrahepatic, it can be due to the liver may not be able to take up everything, or the liver cannot conjugate everything, or maybe most commonly that the liver cannot give the conjugated bilirubin to the gallbladder. That the problem is there. Therefore, the liver will be able to conjugate bilirubin, and because the liver, so like in this case, the bilirubin cannot give the conjugated bilirubin to the gallbladder, not everything at least. Therefore, there will be a lot of conjugated bilirubin in the liver that, because of conjugated bilirubin is water soluble, it will be able to go out of the liver and then through the blood, and then it will be excreted through the kidneys. But also some conjugated bilirubin will be able to reach the gallbladder, and then from the gallbladder it will go through the bile duct to the intestines, and in that case we will get a urobilinogen that will also be able to be excreted through the kidneys. The last type is post-hepatic jaundice, in which the bile duct is completely obstructed, so the conjugated bilirubin, the bile acid does not reach the intestines. Therefore, there will be a lot of conjugated bilirubin in the liver. Therefore, a lot of it will go out through the blood, and then to the urine, or to the kidneys, and then out through the urine. What's important to know here then, that there will be no urobilinogen, no urobilinogen. Yeah. And now I will just talk about what is happening. So this happens in the blood. So this was a seminar task, and our teacher said that it's really important. So let's do it together. But it's basically about the markers that determine what kind of jaundice that we have. So in prehepatic jaundice, in the blood, there will be a lot of bilirubin, but there will be no conjugated bilirubin. And then in the urine, bilirubin, it will not be there because it cannot travel. It cannot travel from the, it's not water soluble, so it will not be able to go to the kidneys. It will not be able to be in the urine. Urobilinogen in the urine will be in high amounts. ALAT and ASAT are biomarkers of liver disease, and prehepatic jaundice does not have anything to do with liver disease, so it will be in normal amounts. And alkaline phosphatase has to do with the bile duct. If there's anything wrong there, then that will be increased, but this should also be normal, or it is normal. Then in intrahepatic jaundice, bilirubin in the blood will be increased. Bilirubin or conjugated bilirubin will also be increased. Bilirubin in the urine, it will be there, let's say present, but the urobilinogen in the urine will be increased. ALAT and ASAT will be definitely increased, and alkaline phosphatase slightly increased or normal. And then bilirubin in case of post-hepatic jaundice, increased, and then conjugated will also be increased. Bilirubin in the urine will be there, but not increased or decreased, but there will be bilirubin there. But urobilinogen, very important, no, nothing is there. This one should be normal because there's no liver disease here, or slightly increased. And alkaline phosphatase definitely increased because there's a bile duct obstruction. Another marker, so this is the last thing I'm going to talk about. The last markers that we need to know is lactate dehydrogenase. In case of damage in the liver, heart, or muscles, we will get lactate dehydrogenase in the blood. Also C-reactive protein, which was the plasma protein. So if we have inflammation in the body due to bacteria, it will be highly increased. And then albumin can also signal for liver disease if we have low albumin levels. And yeah, that's all I wanted to talk about. So